All right, hello everyone. Welcome to your physiology talk of the week on reproduction. The following week, or it might be the week after, we'll talk about pregnancy and we'll talk a little more about these guys. But today we'll start at square one. So let's begin. I hope you are all doing okay. It's a beautiful day outside. All right. Now, this system, the reproductive system, is very different from any other system that we've talked about. There are lots of health problems that can develop with the reproductive system. So that's not unusual. But what is unusual about the reproductive system is it is not essential for life. We can remove these organs and your patients will do fine. And in some cases, your patients will be more healthy and less likely to develop problems. The reproductive system, I really like how the book describes it. It's essential for the survival of the species. And the book talks about just all of the things that have to happen in a particular order at a particular time in order for these systems to work, to produce this cute little foal. You can think of it as a system that is shared by the male and the female. He has half of the reproductive system and she has the other half. I think that's kind of cool as well. My son is taking some sex education in school right now, and I remember how awkward it can be. So there will be some kind of awkward and graphic images. So I'm kind of glad we're doing this remotely. This is a real case I thought I would share with you. Owner brings you his dog Vinny. The owner is really nice guy, but kind of macho, but a nice guy. Think of Vin Diesel or The Rock or something like that. And he's got this really big, tough Doberman who's also actually pretty sweet. But they're kind of a macho, formidable sight, the two of them, right? There they are doing their guy thing out of the park. Why is Vinny being brought to you? Because this is what happens when Vinny gets around another dog. Okay, other dogs try to mount Vinny. This is a little awkward for Vinny and for the owner. Actually, Vinny doesn't seem to mind. So it's really the owner who finds the whole thing kind of awkward. Why are other dogs treating his intact male dog like a female in heat? Well, you'll find out. You will find yourself working with many aspects of the reproductive system. For example, what the heck is going on with that horse's penis? Well, you'll find out, and normally you get to practice doing that in your second year. So let's hope the coronavirus goes away. You'll use anatomy and physiology when you help with this horse and with other patients. You may find yourself doing semen collection or breeding soundness exams or artificial insemination. We'll talk more about that. We'll talk more, you may find yourself working with endangered species or wildlife in other countries, who knows? This is some sort of a rhinoceros they're collecting semen from. It looks like they're very excited. It looks like they've got maybe I don't know if that's lemonade or maybe that's the semen sample there. I don't know. They're very happy though. I'm not sure how happy the rhinoceros is. And learning to identify normal sperm versus abnormal sperm. You'll see various tumors of the reproductive system in males and females. You'll see problems in males. A problem from the urinary system can become a problem of the reproductive system. In the male, the urethra is shared by both the reproductive and the urinary system. And there's actually a stone trapped here. We'll talk more about that. Some dogs have a hidden testicle, cryptorchidism, or a cryptorchid testicle. It just means a testicle that has not descended. Sometimes the testicle is here. Sometimes it's in the abdomen, and the veterinarian has to go looking for it. It's supposed to be here, all right? Not here. And crypt is a root word that means hidden. If you've heard words like cryptic, that's what the word means. An orchid refers to the testicle. This is a case of paraphimosis. The dog's penis is extruded, but it is so swollen and engorged that it cannot retract. What is going on with this cow? Do you see that large pink thing hanging out? That's her uterus, and it is now inside out. That is a prolapsed uterus, and we'll talk more about that next week because that's it's related to pregnancy, or it's related to it's more of a pregnancy and parturition related problem. We see this is a dog that has false pregnancy. She's not pregnant, but she her body thinks it's pregnant. And look, she's trying to, like, you know, she's trying to mother a little rubber ducky or whatever that is. So she's got all these little toys here because she's treating them like her newborn puppies. And hopefully this week, some of you will be discussing some of these different problems so we can learn from each other. Urinary incontinence is a urinary problem, but it has its roots in reproductive hormones, among other things. And again, hopefully we'll be talking about that this week. One reason we like to spay our female dogs and cats is because they can develop a problem called pyometra, where the uterus fills up with, well, I'll just show you. The uterus fills up with purulent material, and it makes the dog very sick. It's toxic. It can kill them. It can rupture inside them. So that is her uterus full of pus. There are some normal things that can look abnormal. Owner brings you a cat that keeps going meow, meow, again and again all night, and is always in the owner's face and keeps its butt stuck up in the air, and the owner thinks its back is broken and it's in pain. You'll find out what's going on. And related to that problem, it's not really a medical problem, but is this normal? Yes. And you'll hear me say the hardest thing to recognize is normal, or the hardest thing to diagnose is normal. And that's why we want you to always be aware of any animals in your life, examine them, examine the farm animals, so you can start to see what normal is. This is a totally normal penis. It has these sharp little barbs on it. Who has a penis like that? Well, you'll find out. And why. So our objectives today, we're going to differentiate between meiosis and mitosis, describe the process of how sperm and ova are made, the structure of sperm, the components of the male and the female reproductive systems, and in the male, some of these accessory structures we'll learn a little bit about. In the female, we'll describe the events that occur during the estrus or ovarian cycle, the various stages, the events that occur. Animals are very different from each other and from humans, so we want to differentiate among the different types of cycles, monoestrus, diestrus, polyestrus, and the seasonally polyestrus species. And on Canvas, I put the student notes, and in general, you can follow along with those notes. There might be some wording differences, but they should be minor. And I'd like to take this opportunity to remind you that at this point, you should be making your own outlines. You should have read the chapter, have your own list of questions that you are hoping to get answered as we study this week. And then you can take my notes and combine them with your notes to make a new set of notes. Now, before we do anything else, you should look at the study notes, and there are questions in the back, the 10 questions. So why don't you, if you haven't already, pause this and go to Canvas and look at those 10 questions and just try to answer them, see what you already know, see what you do not know. It's okay if you don't know any of those answers, but just start, it'll get part of your brain unconsciously problem solving as we go forward. So I'm gonna have a little sip of coffee, you do that. Welcome back. Okay, meiosis. You should have watched the videos on the Start Here page that talked a little bit about meiosis and Hank Green's talk. And I don't want to just read, but I also don't want to skip anything. So I know you can read along with me as well as anyone else. In your body, every single cell in your body, it could be a brain cell, a liver cell, skin cells, kidney cells, muscle cells, whatever. They all have a nucleus, and in that nucleus, you've got chromosomes. Each cell has these 23 pairs of chromosomes. So that's a total of 46. It seems a little strange, but each of those, every cell needs those, even though they don't use every bit of information. At least I don't think they do. They need that because that is the blueprint for you. All of your life functions and enzyme production and how to make this protein and that protein, and everything is encoded in those 23 pairs of chromosomes. And if you were to unravel the chromosome, you'd find DNA and things like that. So but there are two cells in your body that do not have 46 chromosomes. But we'll talk about that in a minute. Again, if we look at these 23 pairs in humans, again, I don't know how one is different from another, but you got all these pairs here. And one pair came from your parents and one pair came from your mother and one pair came from your father. And then one of the actual pairs are these sex chromosomes. So we call one X and one X, and here's X and here's Y. And this male chromosome is kind of short and stumpy, but that doesn't matter too much. I mean, it matters a great deal, but it doesn't really involve today's talk very much. That particular chromosome, of course, is what will determine whether a person is male or female, but it doesn't have much else to do with the reproductive system. So these are these two cells that are unique in I guess any animal, really, almost any animal. There's a cell called a sperm and a cell called an ova, and they are cells, just like a muscle cell or a neuron or whatever. They have one little nucleus, and they are, of course, unique to the reproductive system. The other word for these cells are called gametes, but I'm probably not going to use the word gametes very often, but that's what they are. They're gametes. They don't have two pairs of 23 chromosomes. They just have one set of the 23 chromosomes. And the goal of the reproductive system is to get these two together. What if they both had 46 chromosomes? Well, that wouldn't work, because then they would get together and you would have, what is that, 92 chromosomes. Instead, what happens as they develop, you'll see as we go along here, as they develop, they're going to lead to cells that only have not 46, but one half that number, the 23 chromosomes. And the video you watched in the Start Here page compares mitosis and meiosis. Mitosis is more like a photocopy machine. It's great for making duplicates of the same cell. If I cut my skin, or I bite my tongue, or I get some, or coronavirus damages my alveoli, mitosis will allow cells in those different parts of the body to divide and divide and divide to repair. But meiosis is different. It is not creating copies. It is creating brand new, unique genetic combinations based on the chromosomes that they already have. This N and 2N refers to how many chromosomes there are. So 2N means there's the pair. It's diploid. It's got the the two pairs of 23. 1N means it just has the one set of 23 in humans. Pardon me. Now, I'll be referring to humans here just because many of us are familiar with this idea of 23 chromosomes and 46 chromosomes. But dogs and cats and horses, they have a different number, but it doesn't matter. The same thing applies. The cells are all diploid except for the gametes, the sperm and the egg. They are haploid. They have half the number. Right, just showing you what you would find in sperm or eggs versus every other cell in the body. Just to show you what we talked about, right? If I cut my tongue, or I need to repair some damaged tissue, we can make all these copies of identical cells. But meiosis is different, because now you see we end up with these 4 daughter cells that are all a bit different from these cells. It's shuffling the deck. It's taking these different chromosomes and shuffling them together. And so these 4 could be 4 sperm that were produced, or I would say 4 ova, but you'll see that 4 are made, but really 3 of them are junk, and only 1 is produced, but it's still shuffled. Let me just look at my little notes here. Okay. And like we said, when the two come together, these two haploid cells. Then they form a fertilized egg that we can also call a zygote. And now that haploid number is restored. I'm sorry, that diploid number is restored. Now, the where do all these sperm and eggs come from? So we want to learn these terms spermatogenesis and oogenesis. I might be pronouncing that wrong, but that's how I say it. Um, we'll learn more about the ovary and the testes, but you have um, you know mammals and I'm assuming birds and reptiles. We have these cells called primary spermatocytes in the male and primary oocytes in the female, and they divide through meiosis. They divide into these haploid cells, but they're not really called sperm or eggs yet. It's not until they've divided again that we form what we would call the actual spermatozoa or sperm or the actual ovum here. This figure doesn't show what's happening here. There's actually a you see how there's the four spermatids here. There actually are other um, oocytes formed, but um, they're actually for there are other oocytes formed during oogenesis, but they don't become an ovum. They become what are called polar bodies, and that's there's a reason why. There's a reason why we don't produce as many eggs as sperm, and why do you think that is? Well, one reason is if you think about what's happening um, in the uterus, there are hundreds of thousands of sperm. We don't need hundreds and thousands of eggs. We only need a few eggs. And so that's one reason why spermatogenesis produces so many gametes and oogenesis produces a fraction of that number. The other thing is these polar bodies, I don't totally understand it, but the book describes them as garbage cans for extra chromosomes. Again, I don't really understand that, but I think the point is it's just trying to produce a really good ovum. We can have some sperm that are maybe that aren't the brightest, not perfect. That's okay. We have huge numbers of sperm. We can afford to have a couple of rejects. But when we're talking about the ovum, we don't want to have any rejects. So I think the polar bodies, I think of them as a way of getting rid of any junk, and that we're left with a healthy ovum. So we're going to talk about the male reproductive system first. Again, I want to look at my little notes here. All right. So we're going to look at each of these parts, starting here at the testicle, also called the testes. In fact, give me 1 second here. The so the male portion of the reproductive system, of course, it's going to lead to the development of the sperm. It's also going to be producing the hormones like testosterone in particular. That's really the only hormone we care about in the male. In the male reproductive system, because it's going to play a big role in behavior and physical characteristics, and the other part of the reproductive system of the male, of course, is not just to make sperm, but to deliver sperm into the reproductive system of the female. And so let's meet the sperm. Let's meet the testes. So this is showing a neuter. So there's a dog under that drape, and the surgeon is going to push the testicles forward, or cranially, making an incision, and then they're going to basically kind of pop that out. They're breaking down something called the gubernaculum so that they can get the testicle out the rest of the way. Right, so they're holding the testicle, and then that's part of the cord there. I'm going to clamp that. Okay, so let me show you kind of what you're looking at. So there's this structure called the common vaginal tunic, kind of this, it's like a foot in a sock. Here's the foot, and then that tunic is like a sock that's around it. And when we do a castration, depending on the size of the testicles, we can leave this tunic closed and suture it from the outside, place suture right here. In some really big dogs, we actually open the tunic so that we can ligate parts of the testicle separately. Again, we mentioned the two big functions. We want to produce testosterone. That's one function of the male reproductive system. We also want to make sperm and then deliver the sperm. The parts of the sperm, I think, are kind of cool. Again, it's a cell, right? It has a nucleus. This is a rather unusual cell in that it's like a little tadpole. It's got this little tail on it. It's part of the plasma membrane. It's got this little engine here full of mitochondria. It has this really cool thing called an acrosome, and it's full of enzymes. So when the sperm meets the egg, those acrosomes are going to help to digest an opening so that the sperm can enter the ova, and then they can share their, or they can combine their genetic material. And we learn a little more about that in a week or two when we study pregnancy. The testes themselves are kind of cool. They don't start off in the scrotum. Where do you think they start off? It's kind of surprising. They actually start out near the kidneys. So when a male is just a little fetus, his testicles are up there by his kidneys, and then there's this tough band of connective tissue called the gubernaculum that connects the testicle to the scrotum. And so basically, as the fetus grows longer and longer and longer, the gubernaculum it's not really pulling the testicle into the scrotum. It's hard to describe. It's just that the body is growing away from the testicle. And I don't know, maybe the gubernaculum is pulling it down through this canal called the inguinal canal into the scrotum. The inguinal canal, we all have it. There's just these two small rings. There's one on the right and one on the left. And people will sometimes get hernias right in their groin area. And that's what they're referring to. If they refer to a groin hernia, it's probably an inguinal hernia. Anyway, that's what the testicle has to travel through on its way to the scrotum. And yeah, that's just basically what I said. Okay, you can read that if you'd like. Again, that's gubernaculum. The subtitles are saying gubernacula am. Anyway. And that's the showing you the gubernaculum there. The green arrows are the direction. This gubernaculum is this little structure that was quite long, Well, wasn't quite long. It's just that as the body grew, the gubernaculum held the testicle in place while the body grew longer and larger. Now, why is the scrotum the way it is? You know, why are the testicles kind of far from the body? Why are the testicles not up? Why not leave it up there by the kidneys? Well, I don't completely understand, but sperm do not like body temperature. Sperm like to be a little bit cooler than the body temperature. I mean, for sperm to develop, for spermatogenesis to take place, they need to be a couple of degrees cooler than body temperature. And so that's part of why the scrotum hangs low. It's unique in that there's a muscle called the cremaster muscle that can, in cold weather, it can pull the scrotum tighter and closer to the body so that the testes don't get too cold. And in hot weather, the cremaster will relax so that the scrotum can be far from the body and stay a little more cool. I include John Travolta and the hot tub time machine because there was a minor problem in the 1970s where men were having infertility issues. And they realized that for some men, their testicles were too warm. Men were wearing tight pants, and that was keeping their scrotum too close to their body. And I guess some men were spending all their time in hot tubs, and that was keeping their testicles too warm. And so their sperm, they had low sperm numbers. Anyway, a little history from the 1970s. And this is just to show you, this doesn't show you the cremaster muscle. I don't really have a good picture of that, but it just, again, this slide just tells you what we were just talking about. And it's probably good while we're on this slide just to show you. So sperm will be formed here. They will travel up this structure called the vas deferens, also called the ductus deferens, where they will meet up with the urinary tract, in particular with the urethra, and then the sperm can travel down the urethra through the prepuce. And we'll talk about the prostate gland and things like that, but just so you see how it all kind of comes together. So let's take a closer look now at the testicle and the spermatic cord. The cord, that's what you were seeing here, right? This is the spermatic cord in this person's hand here. Well, that's part of the spermatic cord. Maybe what I should say is it's all in here. I think now part of that spermatic cord, you can also see it running through there. No, I'm sorry, it is all in here because I think that's the vas deferens right there. You'll see. So it's made up of several different structures. There's some connective tissue here that contains blood vessels, nerves, lymphatic vessels, and the vas deferens. The vas deferens, that's what the sperm are going to travel up. And if you've heard the term vasectomy, a vasectomy is where we snip a small part of the vas deferens off and then we ligate either end. And you may find yourself doing a vasectomy. I was asked to do a vasectomy on a ram. I can't remember why, but no one had ever showed me how, but we figured it out. The blood supply, let's talk about that. It's kind of interesting, and the blood supply also contributes to keeping the testes a little bit cooler than the rest of the body. So you have um, you have a you have an artery going into the testicle. And I don't know. We can pretend this is a testicle. We have an artery going into the testicle. Okay, here's an artery. And so that's pretty hot blood going into the testicle. Does that help? So there's very hot blood traveling down this artery into the testicle. Well, then venous blood is going to return. But you have to imagine around this artery, this network of small little veins. It's a very cool meshwork or network of veins. And what they do, the blood coming back will be a little bit cooler than the blood coming in. And so what happens is that cooler blood coming out will cool down the temperature of that arterial blood coming in. And that network of blood vessels is called the pampiniform plexus. Pampiniform plexus. There's a lot of fun words this week. Gubernaculum, pampiniform plexus. So that's that's its purpose. A few other structures we want to learn about. They're in the testicle itself. I think I have a couple of, well, I do have, it's kind of hard to show you this because if you cut into a testicle, it just looks like mush. So we have to sort of use our imagination here. But we're going to learn about things inside the testicle called seminiferous tubules, interstitial cells, and Sertoli cells. We'll learn about testosterone, this structure called the epididymis here. And we already said a little bit about the vas deferens. We'll say a little more. And again, testicle, testes, testes with an I, that's all the same thing. So if we were to cut a testicle in half, it mostly kind of looks like mush, but it's not mush. It's actually thousands of these little tubes, and these tubes are called seminiferous tubules. And what's happening, it's a little hard to, this is a close-up of these little tubes here. I mean, this is a real, like 1,000 times magnification close-up of these little tubes, and these are little sperm being developed by the tens of thousands within these little tubes here. There's lots of these tubes. And so that's where the actual spermatogenesis is taking place. Again, hold on. All right. And it's a little hard to see, but in between here, there's these little areas. In between the tubes, there are these cells called interstitial cells. And their main job is they produce the hormone called testosterone. And the book uses the word androgen. Androgen is to males as the word estrogen is to females, I guess. Androgen just refers to a male hormone. And the only one that I ever hear about is testosterone. So what does testosterone do in men? Now, remember, you make testosterone if you're a woman, you make estrogen if you're a man. Everyone has some amounts of both hormones. In the male, there's more testosterone, and it's responsible for body shape, sex drive, hair on the chest or on the back. It's going to activate spermatogenesis. It'll stimulate the growth of the penis, the accessory sex glands. The prostate's probably the most famous, but you'll see some animals actually have some other glands that are neighbors to the prostate. And testosterone has anabolic effects. And remember, from pharmacology, we learned about catabolic versus anabolic. Anabolic means to build up. So it builds up muscle and bone, and it's influenced by some hormones made by the anterior pituitary gland, LH, and primarily LH, but we'll talk more about LH a little bit later, and we'll talk a little bit about it right now. There are other cells, you can't see them here, so you have to kind of use your imagination, but there are other little cells here called Sertoli cells. And they're also called nurse cells. They provide support, nutritional support, mechanical support to the spermatid, to the spermatid as it develops through meiosis into a spermatozoa. The Sertoli cell also is a shield. Remember, all of these spermatozoa are genetically different from the male. And the immune system will think they are non-self. Remember a few weeks ago we said that's what your immune system does, is it looks at something and says, self, I will leave it alone, or non-self, I will attack it. And so if the immune system were to encounter these sperm, the immune system would attack them. Well, the Sertoli cells protect and shield these developing sperm. I don't know how, they just do. And Sertoli cells excrete or secrete very small amounts of estrogen. So interstitial cells make testosterone, Sertoli cells make small amounts of estrogen. Looking at the outside of the testicle, you'll see it looks almost like a little saxophone or a little hook. And if you look real close, you can see this little coiled tube. That tube eventually leads into the vas deferens. But this whole structure here is called the epididymis. And I call it the finishing school for sperm, or you could call it the high school or community college or whatever you want. When sperm are produced in the testicle, they are not ready to fertilize an egg. There's still some final maturation that has to occur. And so they actually spend, I think, about a week or two in the epididymis before they ever can be ejaculated. So that's the job of the epididymis. And again, there's the vas deferens. Like I said, if you perform, if you help perform a vasectomy, we're making a little cut here and a little cut there, and then we're ligating that. That way, the animal can still make sperm. I'm sorry, well, they can. They still make testosterone, but they're not going to make anyone pregnant because the vas deferens has been cut. Sperm is actually, I'm sorry, semen is mostly liquid, not sperm. So the semen that an animal produces has really very little sperm in it. There's a lot of sperm, don't get me wrong. There's tens of thousands. But most of that liquid is actually made by these accessory sex glands. The most famous is the prostate. I think most mammals, most males have a prostate. Females do not have a prostate, I don't think. Different animals have a few different accessory sex glands, like a horse has this thing called a vesicular gland. Cattle have something called a Cowper's gland. It's not important for us to know the difference. But these glands make and they add other things to this liquid, like fructose and electrolytes and some kind of alkaline fluid. So why do you think that is? Why does semen contain sugar? fructose is just a kind of sugar. Well, it's helping the sperm. The fructose is a food source for the sperm in case they get hungry on their big trip. Um, Electrolytes, the sperm may need electrolytes for their big trip. And the female's reproductive tract is trying to protect itself from bacteria and viruses and fungi and parasites. And so, but it wants sperm to be able to make it. But the vagina and the cervix, they can't really tell the difference between a bacteria and a sperm. And so the vagina and the uterus and the cervix, they tend to be acidic because that kills bacteria. The semen is alkaline, and that will help neutralize the acidic environment of the uterus, so that the sperm don't get killed by that change in pH. Our next stop, as we're heading down the male reproductive tract, is to go down this urethra here, right? So both urine and semen will travel down the urethra. And a little bit of anatomy that's helpful to know is that ruminant and porcine species have this S curve called the sigmoid flexure. Why they have it? It helps them to achieve an erection. They achieve an erection in a slightly different way from stallions and dogs and cats. The sigmoid flexure will straighten out. The S straightens out, and that's how they get an erection. But it also causes problems. Any animal can form a stone. A horse could form a stone. A dog could form a stone. Sometimes the dog or stallion can pass the stone. The stone can travel down their urethra. It might get obstructed, it might not. But in pigs and bulls and bucks and rams, there's a danger because the stone will get trapped at the sigmoid flexure. And plus, I think this is only found in sheep and goats. There's this little structure called the urethral process. Well, first there's the sigmoid flexure, okay, and then there's this thing called the urethral process. The urethral process is at the tip of the urethra, right there at the prepuce. It's this little corkscrew. It's used to penetrate and breed the female, but it's really narrow, and a stone can get lodged there too. It's this urethral process is also called the pizzle sometimes. So this is why we don't feed little goats alfalfa. Alfalfa is it's very rich in calcium and protein, and they can form these stones. And this is even more common if they've been neutered. So a wether, right? A neutered male goat or sheep, is that right? We really would never want to be feeding them alfalfa because they're going to be more likely to form a stone. And traveling down the penis, we've got the outside is called the penis. The tip is the prepuce, and then there's this muscle here called the bulbus glandis, and these are real messages that you might get. You and your veterinarian will be getting so many strange questions, including strange questions about the reproductive system. So this was an actual message. An owner thought his dog had cancer. This has happened more than once. And remember, I said recognizing normal is really hard, or the hardest thing to diagnose is normal. Well, a person can bring you their dog and they have these two big swellings right here. And the owner may think it's cancer. That's the bulbus glandis. That's these paired muscles. Their job is when a dog has an erection, the bulbus glandis muscles are fairly close to the prepuce. They swell up, and that's what locks the male dog into the female dog, so that if she changes her mind, she can't just walk off. He's locked to her, and then he can ejaculate. This is also why some dogs, after they have sex, they're stuck together for a few minutes. That's because we have to wait for these bulbus glandis muscles to lose their blood. I'm not going to talk a lot about all these different muscle layers, the corpus cavernosum and the corpus spongiosum, and they're very interesting. They swell up with blood, but it's kind of hard to explain. I've never really found it all that useful in dealing with health or disease. So we should know a little bit about them, but I'm not going to spend a great deal of time talking about them. But you should read about them. If there's any self-study questions in the chapter, you want to answer those. And here's a little bit. Now here's some applied physiology and anatomy, and this is cleaning a horse's sheath, also known as cleaning stud crud. So let's watch. This is not made by a veterinarian, but I still think it's a pretty high quality video. And it's what is referring to is you're looking here at the horse's penis, and it's within this kind of foreskin here that we'll call the prepuce. So in the horse's actual penis, there's his urethra. There's the part of the penis called the corpus cavernosum. Well, there's this little diverticulum. It's just this little space that waxes and oils that are naturally produced can get stuck in there and form this hard bean, or smegma, it's called. And so we always want to check this. It's also called a sinus or a fossa, but we always want to check this when whenever I'm, doing a dental procedure on a horse, or owners can do this themselves, but some horses require sedation to do it. So let's watch this.
Let's face it, cleaning your horse's sheath is not the most pleasant experience, but it needs to be done. Many people shy away from it completely, but it is an essential part of being a good caretaker to your horse. You will need good old-fashioned water, preferably warm, an epidermis sheath, udder, and genital cleanser. Also, a latex glove, or I like to use a sock turned inside out. This helps to remove the smegma faster. Whichever method you choose, lubricate your hand well with epidermis sheath, udder, and genital cleanser before you get started. You may have to stop while cleansing to add more epidermis to your hands or glove. Sometimes a horse can be violently opposed to having his nether region violated. It's not worth getting kicked or starting a fight with your horse, so save both yourselves the trauma. Bring in the big guns and have your veterinarian tranquilize him for you. Once tranquilized, the penis becomes relaxed and it will drop on its own. Okay, let's get started. Roll up your sleeves and make sure you're not wearing your Sunday best. This is a messy business. Assemble your horse, your hose, sock or glove, and your epidermis sheath, udder, and genital cleanser. And please use warm water if at all possible. If you're lucky enough to have gotten your horse to drop his penis, a cold blast of water will send it packing. Sometimes warm water is all that's needed to get a shy penis to emerge. However, you can still do a good job even if he hasn't dropped. It just requires a little more effort on your part and makes the job messier. Use your hose to get the sheath and its inhabitant wet. If your horse hasn't dropped, turn the hose on semi-slow and insert it inside the sheath chamber. Make sure you have lots of equidermal on your hand, sock, or glove and apply it to the area. Begin washing the penis. You'll feel the smegma and will gently begin to peel it off. If your horse is especially dirty down there, you should apply your epidermis sheath cleanser and leave it on for 10 minutes or so. Smegma can adhere like it has been superglued on, and if you try to pull it off right away, it can be extremely painful for your boy. Let equiderma do its work and begin to melt the smegma away. Now is the time to check for a bean. Gently push back the top of the penis. You'll see the bean, and if it isn't too large, it will slide out with a little help. If it is large, you'll have to get your finger behind it a little and slide or pull it out with your thumb and forefinger. Remember to rinse this area very well after this step. Now all that's left to do is rinse the area thoroughly. Slide the hose up inside and gently jostle it around until the water runs clear and no more chunks of smegma are coming out. This last step will loosen any pieces left behind.
All right, I think she does such a good job, but I think it's funny. I would not wear pearls and a nice sweater while cleaning stud crud. All right, let's see, stop share. Right, and that's where she was cleaning that smegma out. So that brings us back to Vinny. What the heck was wrong with Vinny? Well, if we examine Vinny, I realize this is a little absurd. Vinny had a slightly enlarged right testicle. It was not as big as this testicle, but it was not something the owner would notice. You actually had to feel both testicles, and you would realize that one was just a little bit larger and a little more soft than the other. That leads me to another favorite saying, which is looking is more important than knowing. Looking is more important than knowing. You don't have to know why one testicle would be big and soft, but you need to look. And anyway, so we neutered Vinny and sent in the testicles, and that one large soft one was a Sertoli cell tumor. Now remember, Sertoli cells provide support and protection to the developing sperm, but they also make a little estrogen. And as you'll see, estrogen is the main female sex hormone associated with being in heat, or what we call being in estrus. So Vinny was making a lot of estrogen, and that's why every male dog thought Vinny was in heat, and even Vinny thought Vinny was in heat. So now what we'll do is we'll just do this verbally. We can go and look at well, you'd probably rather just do it on your own. So I will post the answers, and you can just check your own answers on the self-test. So if you want to pause and look at that, you can do that. You don't have to watch me read over it. And I know you'll probably check all your answers. Okay, I don't care. All right, so this is the end of part 1, and then we can stop and go and look at part 2. We'll look at the female reproductive system. So I'll see you there.